Ladies and gentlemen, let introduce me results of field experiment in oak forest realized by the team of Technical University in Zvolen, Slovakia. Evidence of community changes in temperate forest increases. The main drivers of the changes are climate change, nitrogen depositions, abandonment of historical management, and the main effects on plant species compositions are thermophilization, eutrophication, biotic homogenization. Drivers of changes are acting in complicated network of interactions. Moreover, forest stands were affected by different drivers in past and in present. So, is it possible to disentangle causes and their effects? The large effort has been already done by utilization of vegetation resampling, especially by the forest replot group. However, it is predominantly indirect evidence. Therefore, direct evidence is needed, and field experiments can be the promising solution. Aims of our study using field experiment are to simulate effects of some drivers of plant understory change using 3 treatments and their interactions. Our field experiments were set in 2017 in forests dominated by Quercus petra of Carpathian Alliance. There is supposed to be a climax vegetation here in central Slovakia. The area has subcontinental climate, volcanic bedrock, and altitudes were about 500 meters above sea level. Historical management of local oak forests was similar to other regions of Central Europe, including coppicing, cattle grazing, litter raking. One of plots has well-known history due to a historical map from 18th century showing silvopastoral structure. Large trunk remnants of past oak generations still survived in a recent 100-year-old stands. Originally grass-dominated types of oak forests are frequently undergrown by hornbeam and other shade-casting trees and shrubs, and understory is becoming similar to mesic submontane beech forests. In a similar stance we have set our field experiment. Plots of 40 by 80 meters were placed in 5 localities and each split into 8 subplots exposed to different combinations of treatment. Each treatment was applied on a half of subplots. It was, at first, canopy reduction in 2017 when especially shade-casting trees of lower canopy layer were removed. Secondly, annual raking of litter in autumn including removal of deadwood and tree regeneration. And third treatment was nitrogen fertilizing in 2 terms using granular urea fertilizer in dose exceeding critical loads. Vegetation sampling was realized before start of treatments in 2017. Reducing of tree canopy, tree density and removing of lower tree layer, most frequently formed by hornbeam, resulted to open park-like structure. This image is from litter raking happening, realized each autumn after a fall of oak leaves. The next one is showing hand application of granular fertilizer in the beginning of vegetation season on both raked and unraked plots. Canopy reduction and cutting of shade-casting trees resembles past structure of open oak forests. We expect thermophilization, reversing mesophilization and restoring of neural communities. Litter raking simulates also higher export of biomass and nutrients in the past, and it counteracts eutrophication. Here increase of diversity is expected, as opposite negative impact is expected in fertilized plots, including explosion of end-timing bomb in cut plots. The first results show significant increase of herb layer cover in the last year of sampling. However, valid only for plots with reduced canopy, where average covers increased from 19 in 2017 to 34 in 2020. Significant changes were found also in species richness. All localities show similar responses. In some localities maximum richness was reached in third year, in another in the last one. It reached almost 70 species of understory in some plots. However, significant increases were found only when 2 or 3 treatments were combined in plots with reduced canopy. Significant was also shift in species composition of understory, which is apparent already in the second year in DCA ordination space. Among treatments, the highest effect on species composition change was approved for raking by testing interactions with year in redundancy analysis. Raked plots, marked as green triangles, are apparently shifted in the ordination space of DCA compared to original positions of original plots from 2017, which are marked by full circles in a light gray shade. Here you can see the winners, the most responding species to raking and cutting, which belong among nitrophytic and ruderal species, with only one exception of Veronica officinalis, which is more or less oligotrophic species. Better insight into the species composition change in raked plots is provided by synoptic table with frequencies and positive fidelity values shown only for species with significantly higher frequency in particular year. The species can be classified to the group of early colonists, which is consisted mainly by ruderal species and annual species, which mostly missed in the first year of sampling. And the second group, which is composed by species which is mostly perennial, and among them are a lot of target species of nemoral forests, of oak forests, such as Astragalus glyciferus, Latius nigero, Veronica chamedris, which consequently increased their frequencies. Of course, there is a large group of prevailing constant species. Among them, only minor changes can be observed. Some of the species increased their covers, and only Gallium moderatum and Melica uniflora responded negatively by degrees of their covers, by their average covers. Effect of nitrogen addition was controlled by annual soil and litter sampling. However, analysis of total nitrogen content did not show any significant trend in both layers, and annual fluctuations did not follow patterns of treatments, as you can see that fertilized plots show similar pattern in nitrogen content in the litter as plots where no nitrogen was added, and it is similar also in topsoil, like here. Here you can see an example of changes in plots affected by all treatments where was no change in cover, as the cover was high already in original state, but very rapid increase in species richness already in the first 2 years after treatment application. Another example of plot with reduced canopy where understory cover increased while species richness not so much as in rake plots. Understory of this fertilized plot with cut of hornbeams in lower tree layer responded by both species richness and species cover increase. However, the highest species richness was detected in the third year of experiment. The last example shows plot with all treatments where richness reached a maximum of 45 species in the third year of treatment, and the increase was very rapid in 2 consequent years. In the same locality, we observed a rapid dispersal of Poa nemoralis juveniles, established immediately in the 2018 season after the first raking in previous autumn. Here you can see new individuals on bare soil. In neighboring plots covered still by litter, this effect was not observed. Among species positively responding to raking were also some perennials of original oak forests, as Myosotis sylvatica, Veronica chamedris, and Campanula rapunculoides. Falloppia convolvulus established in masses in 2019 in raked plots. It belongs to species with a ruderal strategy, with increasing frequency in resurveyed plots in Slovak oak forests. Difference between fertilized and unfertilized plots was well visible in 2020. Fertilized plants were higher, had darker green shade, and were more frequently flowering. It was similar in the case of Poa nemoralis and Dentaria bulbifera, for instance. Understory of nemoral grasses was reached already 3 years after start of treatments in some raked plots with reduced canopy. On the other hand, understories in plots without canopy removal did not respond significantly, especially when light was shaded by Carpinus betulus trees. Finally, some conclusions could be made after 4 years of our experiment. We observed rapid understory response to treatments, to canopy reduction and litter raking. Fertilizing was less affecting treatment. However, it increases effects of the other treatments when was applied in combination. Significant effect was observed both on cover, diversity and species composition of understory. Rapid increase of species richness with culmination already in 2019 was found in some plots, and dispersal of ruderals and annual species was found immediately in 2018 in raked plots. Target perennial nemoral species show increasing trend, especially in raked and cut plots. They reached maximum frequency in 2020. Among them, Poa nemoralis, Veronica chamedris, Astragalus glyciphyllus, Lathyrus niger, and others should be mentioned, and they represent original species of oak forests. Some nitrophilous, ruderal, and invasive species persisted in communities as well. Falloppia convolvulus meringia trineria represents native ruderals and nitrophilic species, and Erigeron annus solidago canadensis invasive neophytes. Among forest shade-tolerant generalis, Gallium odoratum was negatively affected. Nitrogen addition was not detected by analysis of total nitrogen content, neither in litter nor in topsoil layer. However, effect of nitrogen fertilizing was visible on plant growth and vitality. Thank you for your attention.